Okay, welcome to Chapter 2 on genetics and prenatal development. We're skipping the material from Section 3 of Chapter 2 in your textbook. And, for the genetic material, for some of you, you are really advanced in this knowledge. You're, maybe studying biology or biomedical sciences. And so this is all just very basic for you. And then for others who perhaps were more like me and did not focus on it in high school or in college. In fact, every time I learned about it in middle school or high school or even college, I was pretty much just thinking about what I was wearing and who I had a crush on, and I didn't find it at all very interesting. So for some of you, this is all still a little sketchy, but I'm hoping that many of you were more advanced than me and have actually been paying attention in all of these classes, and that this will be just a quick review. Okay, I've got a little self-study button on this slide. So if any of this is unclear to you, then I encourage you to take a little bit closer look in a textbook, but I'll just hit some highlights here. As you know, we all have 23 pairs of chromosomes in every one of our cells, which makes for 46 chromosomes total. Our chromosomes are composed of our DNA, which is that twisting double helix that we often talk about or see portrayed. And then within the DNA, it's segmented into genes, which then inform kind of our traits. And we have about a total of 20,000 to 25,000 genes, and we'll talk more about that. Okay, so this just shows you a picture of the 23 pairs of chromosomes, and what you can see is that all 23 pairs of chromosomes except for the XY are what are called homologous pairs, and that means that the chromosomes are of the same general size and shape and carry genes of the same type. So then the one difference is XY in males, where the Y is quite a bit smaller than the X and thus doesn't have all of the same genes. But for females, all 23 pairs of chromosomes are homologous. Okay, so the Human Genome Project, it was an international project. It began in 1990 and it was completed 13 years later in 2003, mapping the entire human genome. And so this was, maybe you've heard this metaphor before, this was like taking a book in which there's no spaces and no punctuation between the words and figuring out all the words and sentences. And we were surprised to find that humans only have about 25,000 or so genes, not 100,000 as we expected. So we have about the same number of genes as mice or fruit flies. And so this is just an example of chromosome 9, which shows all the genes on chromosome 9 that are related to different potential traits in our bodies. Okay, so talking more specifically about the human genome, we share anywhere between 94 and 98.8% of our genes with chimpanzees, and I think somewhat similar rates with gorillas. That percentage is a range because it depends exactly on how you calculate it and how rigid you are in saying what's similar and what's not. In terms of comparing ourselves to other humans, 99.9% of the DNA is the same for all humans, so only 0.1% of the genome varies. And of what varies, biological parents and kids share 50% or half. So then what you can see there is that for siblings and parents, only 0.05% varies. So siblings and parents then have 99.95% of the same DNA. And then of course, identical twins have 100% of the same DNA, which we'll talk more about, but that doesn't necessarily mean that all the genes are turned on or turned off in the same way. That's epigenetics. So there can be different ways that genes are expressed for identical twins. So I bet you've learned about this before, genotype, phenotype, right? Genotype is everything that's in your genes, even that is not expressed. And phenotype is what is actually expressed. or observed. And this can be traits like hair color or eye color. When we think about behavior, it could be your behavioral phenotype, which is a phrase that's often used, for example, with people who are on the autism spectrum. It can also be your endophenotype, so what can only be observed through a microscope. But so then your phenotype, it's typically the more dominant genes that are influencing what's expressed in your phenotype. And so we all are carrying things in our genotype, right, that's not being expressed in our phenotype. And so one example here, you can see my husband and I with my son Isaac when he was just a baby, and you can see that our hair is very dark and his is reddish. And this was a shock to us both to have a baby born with reddish hair. But so what that tells us, right, is that one or both of us had red hair in our genotype that was not being expressed in our phenotypes, but went on to be expressed in our son's phenotype. When I was one time sitting with my daughter on the front porch, when she was probably in elementary school and just learned about genotype, phenotype, we were having a little conversation about it. And she said, well, I get it. I must have slow running in my genotype. And she looks at me, but it's not in my phenotype, right? So you might stop for a minute and ponder, what are some things that you're pretty sure, based on your knowledge of your biological relations, of what's in your genotype that's not being expressed in your phenotype. And then the last concept there is reaction range. And so there is a range of what gets expressed in our phenotype, whether that's traits or our endophenotype or our behavioral phenotype, and that it matters based on our environmental experiences. So you may have, we could imagine we all have a range of potential intelligence or IQ. So let's imagine that I have a potential range of an IQ between maybe a 90 and 110, and with 100 being average. So I'm not going to be, even with an amazing enrichment, I'm not going to be a genius, and even in an amazingly deprived environment, I'm not going to be at a really low intellectual functioning. So if I have, better schooling, better kind of activities, support reading books at home, then I might be more at the high end of my reaction range for intelligence, like 110. And if I'm growing up in a family that is more poverty, I have poor schools, my parents are working 2 shifts, they don't have time to read books or can't afford books, then I might be closer to that 90 in terms of my IQ. And that can be the same thing for your risk of skin cancer, like how much are you in the sun, or your musical ability, how much access do you have to musical instruments, or your athleticism, right? How much have you had a chance to practice? So this is just a picture of 2 identical twins in their early 50s, just indicating that over time their genotype, while the same, is now expressed differently in terms of their phenotype. And this is an example of the influence of the environment on how the genotype gets expressed, and also epigenetics, which we're going to talk more about, how some genes get turned on or turned off. Okay, so I've got this marked as a self-study as well. So if any of this still seems unclear to you, then take a look in your textbook to just read things through carefully. But just to hit a highlight here, we generally have pairs of genes to determine each trait, and each part of a pair is called an allele. And you probably remember the Punnett squares that you used to do, the big B, little b, and how if you have the same alleles for a trait, then you're homozygous, and if you have different alleles for a trait, then you're heterozygous, and that typically it's more dominant genes that are expressed. Okay, so once we have heterozygous pairs where we have 2 different alleles for a genetic trait, we usually have talked about it in terms of complete dominance, where only the dominant allele is in the genotype is seen in the phenotype. But it turns out that a lot of traits are actually what's called incomplete dominance, which is an average or blending of the alleles, not one kind of overtaking the other. And so you can see there that a lot of things, for example, around skin color or hair color, could be actually a form of incomplete dominance, as is heterozygous sickle cell, and we'll talk more about that on this next slide. Let's talk a little bit more about the sickle cell trait, which is interesting for a few different reasons. And this often occurs in people of African descent. And so you can be homozygous dominant for the sickle cell trait, so the two big S's there, and that means that you're not going to get sickle cell anemia, but interestingly, it makes you more vulnerable to malaria, and I'll say more about that. Or you could be homozygous recessive for the sickle cell trait, and then you are likely to get sickle cell anemia. Or you could be heterozygous for the sickle cell trait, so the big S and the little s, and interestingly, that is a different phenotype than the other two, and so I'll explain what that looks like. But what that means is that then that kid in the middle is an average or a blend, not just the same as the child who is homozygous dominant. So if it's the whole dominant outweighs the recessive, then the kid in pink and the kid in yellow should be exactly the same, but they're not. So sickle cell anemia, what it does is under conditions of stress, the red blood cells form a sickle shape like a half moon, and that causes them to clot or clog up, which leads to a risk of heart attack or heart failure. And so then if you don't have that, then you're not at risk for sickle cell anemia, death by sickle cell anemia. But interestingly, some of the sickling of the cells somehow is a deterrent to the malaria virus. And so if you don't carry any of the sickle cell trait, then you're more susceptible to malaria. So in some ways, having a sickle cell, being heterozygous for sickle cell, can be protective in that you're not likely to get sickle cell, you're not going to get sickle cell anemia, but you're also not going to get malaria either. So that kid in the yellow has kind of a unique experience, which is optimal in environments where you would be exposed to malaria. The downside of being the kid in yellow is that your cells will sickle somewhat under intense stress, not as much as a child with sickle cell anemia, and this is associated with cardiac arrest. So there's been some research into deaths of college athletes, and some of them are occurring, if they're on like the football field for African Americans in kind of high temperatures and kind of high stress conditions, which have led some of those people who had the sickle cell trait has led some of their cells to sickle and lead to cardiac arrest, even though they did not have sickle cell anemia. And so also, as you've learned, most of our traits are not Mendelian traits where just one gene determines the trait, but most of our traits are polygenic, meaning that multiple genes are contributing to the trait. And so this is just showing an example with skin color and how it's several genes kind of coming together. But this is the same for most things that we talk about, diseases, alcoholism. We can't ever kind of narrow it down. Most of the time we just can't narrow it down to one specific gene. So in terms of how much do our genes determine our traits, which is the standard kind of nature versus nurture debate, and of course we know it's both, how we often figure this out is through two different types of studies, twin studies and adoption studies. And so twin studies are where you compare monozygotic twins who share 100% of their genes, oftentimes with dizygotic twins who share 50% of the same genes. Basically the same as siblings. So dizygotic twins are born at the same time and have more similar environment experiences, also called fraternal twins, but they don't actually share any more of the genetic material than just two siblings because it was two different sperm and two different eggs. Monozygotic twins or identical twins, it's one sperm, one egg, and then the single cell split into two cells and then began forming two different babies. So there's a variety of studies comparing monozygotic twins with dizygotic twins, and also monozygotic twins reared together in the same household versus those that were separated at birth, to try to tease out what's genetic and what's environmental. And we know that monozygotic twins are more similar than dizygotic twins in many areas, even when monozygotic twins have been reared apart in two different households and dizygotic twins have been reared together. So monozygotic twins, for example, are more similar in blood pressure, mannerisms, brainwave patterns, intelligence, some aspects of personality and happiness, and some disorders like anxiety, depression, schizophrenia, obesity, and autism. So certainly genetics can have a part, environment can have a part, but for some things genetics are pretty strong. This is just a picture actually from the Twin Fest that happens in, I think it's Twinsville, Ohio every year, where identical twins from around the world come together for a long weekend and do all kinds of fun things together. And then adoption studies are another way that we figure this out, kind of what's genetic and what's environmental. So for example, there was a adoption study studying the risk of alcoholism. And so basically comparing the correlation between your own or a person's own risk of alcoholism as compared to their birth parents or their biological parents. And what the researchers found is if their birth parents were alcoholic, the person was 4.5 times more likely than others to develop alcoholism. But if their adoptive parents were alcoholic, then they were 2.5 times more likely. So that tells us obviously that both environment and genetics matter in terms of addiction, but genetics has an even stronger influence. So one thing that will come up as we move through the lifespan is that males are at higher risk for death throughout life, even starting as infants, and are also at risk for various diseases or disabilities, more so than females. And so why is this? One of the key reasons genetically is due to X-linked inheritance. And basically, the Y chromosome contains only a couple hundred genes, and the X chromosome contains close to 3,000 genes. And so if you get something undesirable on the X as a male, you don't have anything to cancel it out on the Y. And so kind of evolution takes care of this by naturally humans give birth to more males than females because more males are going to die sooner. Okay, so this is just another visual of X-linked inheritance, and you can see there's all kinds of syndromes on the X that aren't listed on the Y. So things like color blindness, hemophilia, muscular dystrophy, various other developmental disabilities and syndromes. So if you get the short end of the stick as a male on the Y or on the X, you don't have anything to offset it on your Y. And so let's imagine here for another slide on X-linked inheritance, let's imagine the mother is a carrier of an undesirable disease, like muscular dystrophy, and the father is not a carrier. For the daughter, she could, she'll get perhaps the carrier version of the X from her mother, so then she's also a carrier. But if the son born by the same kind of couple gets the carrier version of that allele, he's going to be affected by it because he doesn't have anything to offset it on another X. So this is an important and really fascinating 10-minute episode on epigenetics. And so even if you've learned about it or you haven't, I promise you'll learn some interesting things from taking a look at it and help you understand epigenetics as we move through this class. Okay, so processing what you learned from the film. Can environmental experiences actually alter the gene? And the answer to that is no. Epigenetics is over the gene, so it can turn on or turn off a gene, but it can't give you a different gene than what you already have. Can environmental experiences activate or silence a gene? Yes. So how we are living our lives right now can turn on or turn off our genes. Can that activation or silencing be transmitted to future offspring? Yes, we used to think that epigenetic tags were wiped out and every new baby started with a clean slate, but it turns out that those epigenetic tags can be transmitted to future offspring. So the way that you turned on or turned off your own genes could be passed along. Okay, so just some takeaways from what you learned about epigenetics. Again, epigenetics means over the gene, and it's the use of methyl groups, which are chemical tags. It's one carbon atom attached to 3 hydrogen atoms, and when a methyl group attaches to a specific spot on a gene, a process called methylation, it can change the gene's expression, turning it on or off, dampening it, or making it louder. And then histones can tighten or lengthen the double helix of the DNA that can influence how much the gene is expressed. Again, those bidirectional exchanges between genetics and environment and the heritability of epigenetic tags. So this was mentioned in the SciShow Epigenetics episode. Bygren's research. He was the Swedish nutrition researcher, and this was one of the first and most famous studies on epigenetics. And it actually took him years to get this published because other scientists believed what he was saying was impossible, because they believed that any life experiences basically died with the individual. They did not get passed along. And so the takeaway points here are not to know exactly whether it's boys or granddaughters or grandmothers or girls, but just to know that so oftentimes when a grandparent experienced some sort of drastic change, then in terms of availability of food, then it tended to have some effect on later generations. And so this has made me wonder a lot about our current experience of obesity and how it is not only our current environment, but how maybe it's been influenced by the interactions with food of even, our parents or grandparents. So we're going to be moving into prenatal development, and here's an example of epigenetics in prenatal development. And so if a female experiences domestic violence while she is pregnant, this is actually correlated with an epigenetic change in the glucocorticoid receptor, or GR gene, of her child. Not for her actually, but of her child later on in life. So kids were tested between the ages of 10 and 19, and then mothers were asked if they had experienced any domestic violence while pregnant with this baby. And the GR gene is a regulator of the HPA axis, which you may have learned about, the hypothalamic pituitary adrenal axis that is activated during crisis, fear, and stress. And the glucocorticoid GR gene helps regulate whether the HPA axis is more hyperactive or hypoactive or kind of well regulated. And having an overactive or an underactive HPA axis is associated with mental health issues. And so it's just this idea that perhaps a scary experience and stressful experience that happened to a pregnant mother changed the epigenetic tags of her offspring, perhaps turned on some genes or turned off some genes to help maybe her offspring be prepared for a more dangerous environment. We're going to spend a few slides talking about the differential susceptibility framework, which is kind of one framework within the overall gene environment kind of interaction frameworks. And so basically we know that genetics influence how susceptible people are to environmental factors. And we'd even go further to say epigenetics influences how susceptible people are to environmental factors. And so what the dandelion and orchid hypothesis says is that dandelions are less sensitive to environmental influences, and orchids are more sensitive to the environment, for better or for worse. And so they do better when the environments are really great, and they do worse when the environments are really poor. So basically, just like dandelions can grow everywhere, orchids really only flourish in kind of environments that are ideal for their conditions. Thomas Boyce was one of the developers of the differential susceptibility framework, along with Jay Belsky and Ellis. And so if you have time to watch the whole 18 minutes of the TED Talk, please do. And if not, watch the segment suggested on the slide. So this is another way of viewing the basic model of differential susceptibility. And we're going to look at a lot of graphs in this class that look very similar to this, where you can see that dandelions who are less sensitive to their environment basically stays more the same whether or not their environment is negative or positive in terms of their outcomes. So their red line is more flat, whereas orchids are much more sensitive and reactive to their environments. If their environments are really positive, then their outcomes are really positive, and if their environments are really negative, then they're struggling a lot more. So the orchids are considered highly susceptible or sensitive, and dandelions are considered to have low sensitivity or susceptibility. So there are different reasons for differential susceptibility and sensitivity and different markers of that, and we'll be talking about all of these. We'll be getting some examples from research studies that use all of these types of markers in this class. So the endophenotype or biomarkers, these were things that Boyce was talking about in his video. He was using the example of, he mentioned cortisol, but then the example he used was mean arterial pressure, which is just a form of blood pressure. And kids who had more reactive blood pressure, right, were more orchids. We will also look at quite a few studies that are on genetics and kind of what are called plasticity alleles, genes that are associated with more sensitivity to the environment. We will also take into account epigenetics, the way that the epigenome can turn on or turn off genes and influence their expression, like we just talked about with the glucocorticoid gene and domestic violence. And then we'll also look at studies that use phenotype as a measure of differential susceptibility, with basically babies who are more irritable or more easily startled or more easily frustrated, kind of higher emotionality, as indicative of orchids. These are the genes that have been most studied as far as differential susceptibility, or that are the most relevant to our class. And so the sensitivity or plasticity gene is listed on the left there. And so you can see we have some the 5-HTTL-PR, which we'll refer to as about serotonin, and it is associated with emotion, depression, anxiety, attachment. DRD4 is a dopamine receptor gene. It's associated with attention, motivation, reward, some of those executive functions that we're going to talk about, as well as externalizing behavior, which is like acting out, getting into trouble, and substance use. And then MAOA is an enzyme you can see that affects dopamine, serotonin, and norepinephrine, and it's more associated with aggression. So here's an example of a differential susceptibility using the serotonin gene, 5-HTTL-PR, and it studied whether people had the homozygous short version of the gene, which is SS, and that's associated with being most orchid-like. Homozygous long version of the gene, LL, that's associated with being dandelion, or something in the middle, heterozygous, one short allele, one long allele for the serotonin gene, for this serotonin gene. And this was a study done on a college campus with both staff and students, and it studied, which version of the genes you had, what current life stress you'd had in the last 6 months, and how that was related to your level of neuroticism. And neuroticism is basically anxiety and negative emotionality. And so what you can see there is that dandelions were relatively unaffected in terms of their level of neuroticism by current life events, whether negative or positive. But the orchids, and especially the true orchids who were homozygous short allele, were very affected in terms of their level of neuroticism. And so if they had a really positive and no negative life events, then they had the least amount of neuroticism or negative emotionality. And yet if they had the highest negative life events, then they had the highest negative emotionality. So here's another research study that involved adolescent boys, and you can see that it used a number of different genes, some associated with dopamine and then serotonin and then MAOA genes. So it was studying how many possible plasticity alleles adolescent males had from a group of genes. And so what you can see there is that you could have a range of genetic plasticity, right? You might just have 0 to 1 plasticity alleles, 2, 3, 4, or 5. And so what this graph shows is that for parenting quality, the connection between parenting quality and level of self-control or self-regulation, for the dandelions who did not have any mini plasticity alleles, their behavior was not so predicted by their parenting quality. But for the orchids, those who had the most plasticity alleles, their behavior was really predicted by the parenting quality. If their parenting quality was low, then they were the most poorly behaved, and if their parenting quality was high, then they were the best behaved. So this was a meta-analysis of many intervention studies as related to differential susceptibility, or specifically genetic susceptibility. And what they found is that overall interventions, whether that's educational programs or parent training programs, were more effective for individuals who were more orchid-like, who had more genetic plasticity. There were greater differences for interventions that were targeting externalizing behaviors, things like substance use or violence, than internalizing, which is anxiety and depression, and bigger differences between the orchids and dandelions if you were looking more at dopamine genes versus serotonin genes. But the takeaway from this is that even though orchids are going to be more affected by life events, negative or positive, they are actually going to be more affected by intervention. And so it may be the orchids often who benefit the most from interventions. So you've probably already been wondering, which one are you, a dandelion or an orchid, or maybe something in the middle? And how maybe you can figure this out a little bit is how much your well-being changes over time depending upon what's happening in your life. So I think of myself as more of an orchid because when I was a kid, my well-being, how I was doing, really went up and down with how my parents were doing. When my parents were highly stressed, I wasn't doing so well. And when my mom went back to school and became a marriage and family therapist, I started doing dramatically better. So I think those of us who are more orchid-like are more affected by what's happening around us. So in terms of what this means for clinical work, I do think it's probably the orchids that keep therapists in business. And so if you're an orchid, you can feel a little bit bummed by that. And again, Boyce was suggesting that maybe about 20% of people in his samples were coming out to be orchids. So if you're more orchid-like, then you are going to experience the world in a more intense way, for better and for worse. The bad news is that tough things might hit you really hard, but the good news is with support or with intervention, you can end up really being a rock star. But this kind of explains to us why some of us have such a hard time with things, and those of us who are more orchids might be asking, like, what's wrong with me? Everybody else isn't having this big of a reaction. Everybody else seems more okay. Why do I have to be so intense? And so a lot of it is probably in our genetics, and maybe in combination with our early life experiences, our epigenetics. Okay, so now we're going to switch gears into conception and prenatal development. And I do have to say just a couple of disclaimers. One is I often will say you when I refer to getting pregnant, even though I know not all of you will become pregnant or want to become pregnant. And then secondly, around sex and gender, I will try to use female and male because I know that, for example, someone could be male and identify as a mother and female and identify as a father. I'll also try to use terms like pregnant person rather than pregnant mother, but I will default sometimes to she, mother, her, women, only because all of the research so far is focused on cisgender women who are pregnant. Okay, so I bet you've learned about mitosis and meiosis in the past, right? Mitosis is normal cell division, and meiosis is the formation of gametes, which are sperm and egg cells. And the main thing that I would want you to know here is that through the process of meiosis, it results in 4 gametes. In males, all 4 of those become sperm. In females, only 1 of those become an egg, and the other are just polar bodies that eventually degenerate. And then the other piece to know is that gametes only contain 23 chromosomes, so no pairs, right? Just a single version of each of the 23 chromosomes, so that at the time of conception, when a sperm and egg come together, then there can be a combined 46 chromosomes, or 23 pairs. All right, stages of prenatal development. There are 3 of them: the germinal period, the embryonic period, and the fetal period, and we're going to be talking about them more in depth. But first, the 2 important things that I want you to know. You can see that these periods are not divided into equal amounts of the pregnancy. So they do not correspond to trimesters. If you've heard people talk about trimesters of pregnancy, that's a third, a third, a third. But what you can see here is the germinal period is only 2 weeks long. The embryonic period is 6 weeks long, and the fetal period is the rest of it. So that's, about 32 weeks long. The other important thing to know is that the way pregnancy is referred to in terms of weeks by doctors, by OBGYNs, is different than how we are referring to it in our textbooks. So doctors talk about your pregnancy as 40 weeks. But pregnancy is calculated based on the first day of your last menstrual period, because it's harder for females to say when they had sex and when they got pregnant, and we don't know that as precisely, so we ask females when they had their last menstrual period, and that's how we calculate pregnancy. But conception usually happens 2 weeks after the first day of your last menstrual period. It's kind of in the middle between periods. So basically, by calculating pregnancy that way, there's actually 2 weeks where nothing actually was happening. There was no conception taking place. There's kind of a dead window there. So even though we say pregnancy is 40 weeks, it's actually only about 38 weeks from conception to birth. And so anytime we are saying in our textbook, we're saying 2 weeks pregnant, that the doctors would actually be saying that's 4 weeks pregnant. They'd be adding in that extra 2 weeks. So if you're kind of comparing what you're reading in a textbook with pregnancy books, you have to be adding 2 weeks into what we're hearing in our textbooks to match up to the pregnancy books. So this video from your textbook, Key Events in Prenatal Development, is really important. It's a little lengthy, about 6 minutes, but it provides a great overview of prenatal development from conception to birth. It also describes the importance of maternal health and how teratogens can negatively affect baby's health. And we'll talk more about teratogens. So let's talk more about the germinal period, which is the first period of prenatal development. It's 2 weeks long, so from the point of conception till the end of week 2. And during this period, we call the developing baby a zygote. So we actually change what we call the baby in each prenatal period. And so in the germinal period, we call it a zygote. So basically, the sperm and the egg have met in the fallopian tube, not the uterus. Conception usually takes place in the fallopian tube. And so now we've got the 2 gametes that have combined to form a cell with 23 pairs, or 46 chromosomes. And so then that cell begins migrating through the fallopian tube down into the uterus. And after about a day, it divides itself through mitosis. So now we have 2 cells of 46 chromosomes each. And it keeps doing that. So around a week, we have a blastocyst, which is a hollow ball of about 150 cells. And we've got some more pictures coming. And that blastocyst is going to begin to differentiate itself into the embryonic disc, which is going to become the embryo or the baby in the next phase of development, prenatal development. And then the second part is the trophoblast, which basically becomes the house for the baby. And so the trophoblast begins differentiating into the amniotic sac, the placenta, and the umbilical cord. And I know we usually talk about the amniotic sac and the placenta as if it's the same thing, but the placenta is actually a pancake-shaped organ that's attached inside the one side of the amniotic sac, and then the amniotic sac is the whole thing surrounding the baby. The germinal period is marked at the end once the zygote implants into the wall of the uterus. This is just a picture kind of showing the process that I just described on the previous slide of where conception takes place, and then mitotic cell division, forming of the blastocyst, and then implanting of the zygote. These pictures just portray the differentiation of the blastocyst that I talked about. So the blastocyst is the hollow sphere of about 150 cells. And then you can see that the outside of that sphere becomes more the trophoblast, or the house for the baby. And then there becomes this clump of inner cells, which is the embryoblast, which is going to develop into the baby.
So now we've reached the embryonic period, which is a 6-week period lasting from the start of week 3 to the end of week 8. And now we call the developing baby an embryo, not a zygote. And this is once the zygote again has implanted in the uterine wall. This is the time when most people discover that they're pregnant, or about the earliest you could discover that you're pregnant. And it's also a time when all the major organs, with the exception of the sex organs, develop, even though they're tiny. It's a time of rapid cell division and differentiation, where cells are acquiring kind of their own specialized structure and function. So some cells are going to go on to be skin cells, and others are brain cells, and so they're all sorting themselves during this short period of time. So by the end of the 3rd week, or the very first week of the embryonic period, there are 3 differentiated layers, and these all turn into specific things. Once a cell has become part of the ectoderm, it can't turn back and change to the mesoderm, right? It's only moving forward in that pathway. So the embryonic disc turns into 3 layers. The ectoderm, which is going to become the central nervous system, the neural tube, the brain and spinal cord, the sensory organs, the skin. And it's the fastest of these to develop. Then the endoderm, that layer will become the stomach, liver, and lungs. And the mesoderm is going to become muscles, bone, reproductive and circulatory systems, including the blood. So this is just a picture showing you what it looks like when the embryonic disc transforms into 3 layers and the ectoderm folds in on itself to become the neural tube, which again will be the brain and the spinal cord.
Continuing on with brain development in the embryonic period. So after the ectoderm thickens and folds and the neural tube forms, then we've got neurons that are beginning to differentiate, some going to form the brain and some going to form the spinal cord. And as early as week 7, we'll start to see some axon and dendrite connections begin to form between neurons. Here's some other things that are happening during the embryonic period, that 6-week period from week 3 through week 8. In addition to the central nervous system coming online, all key organs, with the exception of the sex organs, are forming, and this is called organogenesis. And so we are seeing the heart form and begin beating, eyes, nose, and mouth have formed, bones, or the skeletal structure becomes in place by the end, with arms and legs, and the digestive system is formed, including kind of working kidneys and liver. And this just shows even the change from week 3 through week 8. this little thing is only about a grain of rice, the size of a grain of rice in week 3, and then an inch long by the end of week 8. Yet by the end of week 8, you can see hands and fingers, all key kind of body parts. So the prenatal video in the textbook that you watched talked about teratogens. Teratogens are basically anything that could be harmful, something external that could be harmful to the developing baby. So it can, as we're going to see, it can be substances, but it can also be things like stress. And so this graph is a little bit hard to understand, but I want you to notice that it breaks out the age of the baby in weeks, but it lists every week up until week 9. It has a column for each week up until week 9, and then a column for week 16, and then basically 2 columns for the whole second half of the pregnancy. And the red bars indicate the most critical period of basically organ formation, when the organs could be most susceptible to teratogens, and then the yellow bars indicate kind of still a cautionary period, but not quite as critical. And so the key takeaway points here are that teratogens are the most dangerous during the embryonic period. You'll notice that teratogens don't have any effect during the germinal period, during weeks 1 or 2. What happens, babies do often, zygotes do often fail, or babies die during that time. It often just appears to be like a heavy period, essentially. Most of the time, females aren't even aware that they were pregnant if a baby is to die during those first 2 weeks. And it's often due to some sort of genetic defect or chromosomal abnormality. So you don't have to worry about like, oh my gosh, I drank a whole bottle of wine, or whatever, and I didn't even know I was pregnant for those first couple weeks. Generally, that wouldn't have much of an effect, because you don't actually find out that you're pregnant. What they'll often say is you're 4 weeks pregnant, but you're usually just 2 weeks pregnant. That's the earliest we can find out, is at the end of the germinal period. So usually about the time you find out you're pregnant is when you would maybe change your habits. And so it's good to know that if you're kind of on top of it, you'll know in time to kind of change what you're doing. So basically, we're going to be going through teratogens and their influences. And the interesting thing is we have to be most careful about them when a lot of times nobody even knows that this female is pregnant. We worry a lot more about pregnant people when they're really big and it's obvious, and don't stress them out, and let me get that for you, and let's eat healthy and stuff like that. But we really should be worrying the most at the very beginning. So we'll talk more about it. Okay, so once we finish the embryonic period, we're on to the fetal period, which again is the longest period. It's week 9 through birth, which would be 38 weeks gestation, or as they would say, 40 weeks pregnant. It's going to be the end of the first trimester, and then all of the second trimester, and all of the third trimester. And so you remember that most of the organs, except the sex organs, developed during the embryonic period. And so now most of the rest of the pregnancy is just about growth and maturing of the organs and development of the sex organs. At about week 9 to 10, the heart can be heard from the outside with a little microphone. Before that, if you want to know if a heart is beating, you have to look at it on an ultrasound, because it's too small for you to hear it with a microphone up to the person's belly. Babies begin during the fetal period to adopt common sleeping positions and a sleep-wake cycle. Usually the first fetal movement, which we're going to talk more about, is felt around week 13. Around week 20, the sex of the fetus can be determined visually, so that's often on that 20-week ultrasound that if people want to find out the sex of the baby, they can. Near the end of the second trimester, babies are opening and closing their eyes, sucking their thumb, responding to light and sound. And by the end of the third trimester, their heart and lungs are increasingly able to sustain life. Lungs are the last organ to mature. Even though they develop during the embryonic period, they aren't ready to breathe air until right near the end. And so when babies are born prematurely, that's often a challenge. Babies continue to gain weight and length. During around the 7th month, they begin to turn upside down in preparation for birth. Okay, in terms of brain development during the fetal period, more of it is focused on the cerebral cortex, which you may remember is the outermost layer of the brain. So the outermost layer of the brain, the neurons are being created, and we're starting to see connections form in the cerebral cortex. Okay, so around week 13 or near the start of the 2nd trimester, the fetus movement begins to be felt. And initially, pregnant people will say it just feels kind of like a butterflies, or it almost feels like indigestion. You're not even sure if you felt the baby moving. But that feeling just kind of gets stronger and bigger until eventually, right, you feel really big movements, and you might even be able to see kind of body parts like elbows or butts or sticking out, or trying to figure out kind of what they are. Interestingly, prenatal activity, how much a baby moves around in utero, predicts how much this baby is going to be physically active, like at age 1. So a really active baby does mean a really active toddler, typically. And then movement increases, but then once we get near the end of pregnancy, things get really tight in there, and then movement gets more challenging. So I encourage you to watch the first 4.5 minutes of this video, which basically shows prenatal care across cultures and around the world. Okay, so these maps show access to prenatal care by race and ethnicity in the United States. And so darker colors means late or no prenatal care, and light colors means better access to prenatal care. And so what you can see is that across the U.S. For women of color, there is on average more likely to be later or less access to prenatal care. And for many years, this was explained basically in terms of income, that it was more about income than it was about skin color. But kind of recent conversations are starting to kind of call that into question. And so we've got more information coming about that. So I encourage you to watch about 5 minutes of this TED Talk, watch from about 6.5 minutes to 11.5 minutes by Miriam Zola Perez, that's on Jenny Joseph and the JJ Way, and that's Jenny Joseph kind of pictured here. And the idea is that racism basically is a contributing factor to why persons of color might not feel comfortable seeking prenatal care, or might not have as high quality prenatal care. And we might see similar patterns also for transgender people who would be more at risk for not getting kind of adequate prenatal care. So in terms of diet while pregnant, there's some kind of key things. So folic acid or folate is a really important one for preventing neural tube defects like spina bifida, and thus also miscarriage. And folate naturally occurs in many green vegetables, citrus, beans, nuts, and it has been added to grain products in many countries. So if you look at a bag of flour, it now will say folate, and I think maybe even into orange juice, for example. But before this, lots of women were more likely having miscarriages, and particularly rural women whose babies are conceived in the winter and aren't getting access to kind of fresh fruits and vegetables and don't have fortified flour are more at risk for neural tube defects and miscarriages coming along with those. And I knew someone who, kind of an extended family member who had many miscarriages in the late 1960s and early 1970s before they realized that it was because she didn't have enough folate. So we'll be talking more about prenatal vitamins, but that has been a big source also of getting adequate folic acid or folate. Okay, so iodine is another deficiency. We iodize our salt in the United States, so we are less likely to be deficient in iodine, but that happens more in developing countries, and again can result in miscarriage or brain abnormalities. And then iron is also necessary, and that's generally from green, leafy greens, red meat. And it's necessary to prevent kind of premature or low birth weight. Iron is another thing that's added to prenatal vitamins. The challenge of adding iron is that it can also be constipating, and so some women can't tolerate the iron in their prenatal vitamin. In general, even though the research on vitamins is kind of multivitamins is up and down, I think right now, generally people are pretty supportive of prenatal vitamins for both females and males who are either getting pregnant or getting someone pregnant. So what is recommended is that you don't just wait until you're pregnant to start a prenatal vitamin, but if you're trying to get pregnant, you go ahead and go on a prenatal vitamin so that you've got kind of everything that you need before you even conceive, because a lot of these things like folic acid are necessary, before maybe you even know, or, very soon after you would even possibly find out that you're pregnant. And prenatal vitamins have been shown to improve the quality of sperm as well. So it's a good idea for males too. All right, so let's talk about exercise while pregnant. So it used to be that they thought exercise wasn't a good idea when females were pregnant, but now we know that continued physical activity is generally a good idea because aerobic exercise increases the flow of oxygen through the mother's body and also to the baby as well. The main concerns are to avoid any exercise that could risk injury or falling, so, no rock climbing kind of thing. Also, you have to be careful about exercise that would cut off the blood supply or the oxygen to the fetus. So concerns about that particularly happen when you're in the second part of the pregnancy, so around 20 weeks. You can no longer do exercise lying on your back. So if you had been, for example, going to the gym and doing some bench press, that's when you would have to stop that. You also, if you were going to a Pilates class that was really on your back, you'd have to move to kind of standing Pilates exercises. And you're also not supposed to be doing heavy lifting above 10 pounds as you get into that second part of pregnancy, or that last half of pregnancy. The other risks are you don't want to get your body temperature, get too hot, or your blood pressure to get too high, because that could also have negative effects for the baby. So you're basically, you can still be running, but you're probably running pretty slowly and, not pushing it so hard. It isn't the time to start new activity that you hadn't been doing. So if you weren't running before you were pregnant, or you weren't weightlifting before you were pregnant, maybe now's not the time to start doing it. I actually ran through both of my pregnancies. What stopped me in my first pregnancy about 5 weeks before Isaac was born was that the bones in my feet grew, which can happen. I think it's because of the hormones, not because of the weight, and my feet hurt so much I couldn't run. But in my second pregnancy, I ran very slowly, just about 2 miles, up until like 3 days before Iris was born. One challenge with running is that the progesterone can make you feel more short of breath, and also the hormones can make you have to pee even before the baby gets very big. And so I used to say we should have like a pregnancy potty 3K, where you run 3 kilometers and there's a bathroom like every kilometer. Let's talk some more about specific maternal teratogens that have been studied. So on this slide, I'm going to talk about a few, and then I've got some upcoming slides that will go into more detail. So on this slide, we're talking about malnutrition again, and we basically alluded to this when we talked about the importance of diet in prenatal care, but malnutrition can be considered a teratogen, not getting adequate kind of nutrition is obviously harmful to a developing baby. Infectious diseases have also been shown to be harmful. So things like the flu has been shown to have kind of a later increased risk of schizophrenia. So this is why if you were thinking about getting pregnant, you'd want to get your flu shot. Measles can be a risk. Alcohol is more of an issue in developed countries than developing countries. And it's interesting how the research has changed even in the period of time that I was having kids. So they used to say, oh, in moderation, maybe up to a drink a day. And now I think we're realizing that it's really better to just not do any. Even 1 or 2 drinks, a few days a week puts the developing child at risk for lower height, weight, and head size at birth, and for lower intelligence and higher aggressiveness during childhood. So take it from me, who had a glass or 2 of wine during my first pregnancy, because that was 20 years ago and people were like, oh, it's no big deal. Once you start having, all kids have glitches, right? We all have our quirks. We all have things that aren't perfect. And as the person who, created them essentially with your own body, you will think that those quirks are your fault. And so then you'll wonder if it was that glass of wine you had at your uncle's birthday party. So you might as well just skip it. Okay, so caffeine is similar. It used to be like, oh, you could have a couple cups of coffee, and some people will still tell you that you can, but basically even 150 milligrams of caffeine can lead to spontaneous abortion and miscarriage. And so we're really suggesting that you try to limit caffeine during pregnancy. Okay, so then on our upcoming slides, we're going to talk about obesity, nicotine, cannabis, and stress. So more attention is being paid to the negative effects of obesity on the developing baby. Obese mothers are more likely to give birth to babies with all of these types of health outcomes: obesity themselves, diabetes, high blood pressure, heart disease, asthma, autism, ADHD, and developmental delays. And it's been hard to figure out what's about, just lifestyle. So if a female isn't eating as healthy in her life, then and this is leading her to, and not exercising, this might be leading her to be more obese. And then that leads, those lifestyle factors lead her baby to not have as good health outcomes. Or is it more likely about genetics, or is it something more about kind of physiology? And so there's definitely research, that genetics kind of passes along obesity risk. And there's certainly research that says lifestyle factors matter. But there's also research that says that when a woman gains too much weight, or when a female gains too much weight, it actually changes her physiology and maybe her endocrine system. And this then influences the baby's physiology, which can then put them more at risk for some of these challenges. So generally, nicotine is known to be a negative when you're pregnant, and it's shown to have kind of higher risk of miscarriage, premature birth, low birth weight, also later risks of lower language skills, lower attention and memory, or behavior problems. Secondhand smoke has also been shown to have some of those effects. But if we return to the differential susceptibility research, this was based on the DRD4-7R gene, which is associated with dopamine, and you can see that for the kids who had one or more of the plasticity alleles, they were much more likely to be negatively affected by the smoking of their biological mother. So if she smoked, then they had a much higher risk of ADHD, and if she didn't smoke, then they had the least amount of risk of ADHD.
Okay, we always get asked about cannabis, and some of you may have had biological mothers who used cannabis when pregnant, like any of these teratogens that we're talking about. And there's I'm citing a few different research studies here, so I can't tell you the specifics on like how much or how often, but I will say it seems like most studies were investigating higher use or use over multiple, not just at a single time, or even in a single gestational period. So maybe you're using it, both in embryonic and the fetal period. It seemed like most of the effects were found when people used, the pregnant person used cannabis in 2 or more periods of pregnancy. Now, what I don't have is any research on biological fathers. This is only biological mothers. And so you can see the short-term correlates here. congenital abnormalities, kind of otherwise known as birth defects, stillbirth, so a baby dying in the second half, the last 20 weeks of pregnancy, being born prematurely, being born low birth weight, having to spend time in NICU or neonatal intensive care, having a small for gestational age or small for date age size, meaning that even after you're born, when you're 2-month-old, 3-month-old, you're smaller than usual, and higher risk of a baby dying in the first year, which is known as infant mortality. Long-term correlates, conduct disorder, and I can't tell you the ages on this. This is kind of a review study. Delinquent behavior, depression, this is in kids, the children, substance use disorder in children, anxiety in children, and lower attention or memory scores.
So maternal stress is considered a teratogen, and there's been a lot of research on maternal stress, experiencing, for example, the death of a family member or domestic violence or, a crisis like Hurricane Katrina during pregnancy. And there's not clear kind of research that says experiencing a stressful event in the first trimester or third trimester or second trimester is better or worse. As you might imagine, I, for example, might imagine that it's worse to have stress during the embryonic period because that's the most susceptible for teratogens in general. But maternal stress shows basic kind of findings across all trimesters. So this was a research study that involved basically national registries among Danes. So in Denmark, over 1 million Danish births between 1970s and 1990s were tracked and then correlated with whether or not the pregnant mom had experienced the death of a close family member while she was pregnant. And then what that led to for mental health diagnoses later on. And so you can see that experiencing the death of a loved one in the first trimester put children later at more risk for schizophrenia. In the third trimester, it put them later at higher risk for being on the autism spectrum or being diagnosed with ADHD, which also was the case for experiencing maternal stress right before conception.
So there's a pretty large maternal stress during pregnancy literature, but it's hard to kind of come up with the perfect, recipe of how much stress is too much. I would say most studies are exploring moderate to high levels of stress. Some studies, for example, have focused on intimate partner violence or pregnant females living through natural disasters. However, as we'll see, it can be lower to moderate amounts of stress too. Some of these also include anxiety and depression for the mother. And what I can tell you is that even though we talk about teratogens being more crucial during the embryonic period, maternal stress literature does cross all gestational periods, so germinal, embryonic, and fetal. And we're still figuring out what the different effects are, like you saw kind of on the slide around, experiencing the death during different parts of pregnancy. On average, higher maternal stress is associated with shorter pregnancies, babies being more likely to get infections in their first year, having delayed or lower motor development, cognitive development, and that's more associated with stress during the first half of pregnancy. A more difficult infant temperament, and then this is a more emerging area of just system changes in the baby's biological system, like decreased immune system, disrupted gut microbiome, or reduced telomere length. And yet, it's not always just severe or moderate to severe stress. It can just be anxiety about pregnancy, which kind of is a distinct syndrome defined as kind of having fears about the health and well-being of one's baby, childbirth, hospital experiences, or being a parent. And so what you can see is that the more a pregnant female has that, the lower infant mental or cognitive and motor development and at 8 months, and higher preterm birth. And again, this is correlated, so it could be that the pregnant female has some reasons to worry, and that's just correlated with premature birth, but those things are correlated. And so I think the bottom line from all of this is stress is going to be inevitable during pregnancy. In fact, just reading and learning about all this maternal stress literature always stressed me out. And, oftentimes stress is unavoidable, some stress, but if we are experiencing more stress, then we have to kind of increase our social support and up our coping skills. I, for example, moved and started a PhD program when I was halfway through my first pregnancy. I defended my dissertation with my second pregnancy. I was certainly pretty high stress, and yeah, sometimes I wonder if that led to some of my kids' quirks. But overall, they turned out okay, despite me being a fairly stressy person.
So let's return again to the differential susceptibility research and how it relates to maternal stress during prenatal development. So even though we make kind of these general statements that maternal stress can lead to negative outcomes for kids, we can see here that once again it can depend on whether or not you are more genetically an orchid or genetically a dandelion. And so this was studying the DRD4 gene again, which is associated with dopamine, the same one as the smoking graft. And you can see that it was the level of maternal stress while the female was pregnant. And then on the left-hand side, it's the child behavior checklist, externalizing behavior, sometime between age 8 and 15. And so this is kind of acting out behaviors, getting into trouble at school, getting into fights, problems with peers, getting into trouble at home, those kinds of things. And so what you can see is that maternal stress really didn't have a big effect for the dandelions, but it had a significant effect for the orchids in that if they had a mom that had a high amount of maternal stress, then they had the highest amount of problematic behavior, and if their mom had the least amount of maternal stress, then they had the lowest amount of problematic behavior.
Okay, so we focus so much on the biological mother and her exposure to teratogens, and there's really just emerging research on the biological father and their exposure to teratogens. And what you can see here is and again, I think this is always research you want to circle back on should you or your partner trying to get pregnant. But there is evidence that basically says, that there is lower sperm quantity viability, which is basically the sperm's ability to penetrate the egg, and DNA quality associated with a number of things that I'm going to talk about here. So when you think about teratogens, the difference is that for the biological father, this all happens before conception. It's not obviously while the biological mother is pregnant, because the sperm has already been released. It's already done its job. Teratogens for the biological father are all about what's been happening prior to conception that could influence the quality of the sperm. So there is some startling research about cell phones. Obviously, this must not be hugely damaging because kids are still being born okay, but it does appear that cell phone waves are related to DNA fragmentation, and nicotine is related to the kind of viability of the sperm, also the motility, basically how fast those sperm can swim, and also zinc levels. Anabolic steroids can reduce the number and quality of sperm. Cycling, interestingly, and I think this is not so much like biking to class, but biking for many hours. So yeah, if you're trying to impregnate someone, this might not be the time to be spending lots of time in the bike saddle, because that's creating some challenges for the sperm. And then endocrine disrupting chemicals, this we start to hear about in the news media. These can be things that people might be exposed to in pesticides, sunscreen, plastics. These are both for biological mothers and fathers. So we're always trying to be aware of our environmental hazards. Alcohol, I've really just so far seen evidence that it disrupts DNA methylation or epigenetics in mice. I haven't yet seen it in humans, but it might be out there. Prescription medications, when I was looking at this most recently, it's like a lot of them are no, but I think that have been tested, but I think, if you're, again, hoping to impregnate someone, then it's worthwhile to kind of ask whether or not you should be on your prescription medications. And again, like I mentioned previously, taking a prenatal vitamin as the biological father prior to getting someone pregnant is a great idea. You can test yourself here with this question: what's the most common harmful teratogen worldwide? So this is global. And so the answer here is malnutrition. If we're looking at the whole world, because most people live in developing nations, they might be experiencing, some of which are experiencing malnutrition, not all, obviously. In developed countries, alcohol is the #1 teratogen, followed by tobacco.